and soon I will share with you the YouTube link in the link page so everybody can watch. Here is the link. And update it. So we are live on YouTube. Welcome everybody. We're recording also. And I can share now my screen with the presentation. Here it is. It's official. We started. Welcome everybody. This is the 2020 Padova BrainHack. We called it BrainHack Diversity, Inter-Individual Variability in Cognitive and Clinical Neurosciences. Is this a signal or a noise? This title is very long and it started from an internal discussion amongst the organizers that you can take part of. Is the variability a noise or a signal that we can have to deal with? We will discover together. What is a brain hack for newcomers, people who haven't never been to a brain hack? It is an academic meeting format in between a collaborative hackathon and conference and tutorial for newcomers. So if you are unexperienced, it can be a very good occasion to learn something new for you. The event format is thought to encourage the participation as much as possible, regardless of your experience and formation. BrainHack benefits and collaborates to open and reproducible science. There are tons, literally tons of open source code and open data which is available online for everybody, and you can produce such data and such code for everybody in the future. And also around the BrainHack, it has been formed a global community. So a BrainHack is a global event, which now is this year in all continents, as far as I know, except for Antarctica. And you can check out, to reach the community, the BrainHack Mattermost channel, which is the official channel of BrainHacks with hundreds of thousands of people here. So how to BrainHack, just for newcomers. As first, enjoy the talks. The Ignite talks are meant to inspire participants for the working group. So be inspired, be amazed. Choose between the project. This year we are very lucky and we have 8 great projects you can join. And join the working group and work on a project and have fun and learn with the working group. Talking about the working groups, we have a novelty from this year. You can submit and publish your result in the BrainHack proceedings. This is an open source, an open access and free to publish journal for the outcomes of BrainHack projects. Of course, you are not forced to, but if you want, you can think about publishing your results. Of course, these are a brand new publication, so it's not Scopus indexed journal. It has not at this moment all the characteristic of well-established journal, but it's free and it's open access. So think about it, and it's also peer-reviewed. Still talking about working groups. This is a proposal. You can create a GitHub repo of your working group and project and share your creation with the world. So this is the gathered BrainHack PD 2020 repo that we created for this event, and we will link, if you want to, your GitHub repo in this. If you're not familiar with Git and GitHub, do not be afraid. We're here to help you and we can help you sort out things. So call for help. We are here for you for all the 3 days. Please, all the participants shall read and accept the code of conduct, COC. And please behave. The code of conduct is very simple. And if you think that some people have broken the code of conduct against you, please report it to the organizer and we'll take care of the issue. In a nutshell, this is bad and this is good. So don't plot to kill everybody and simply have fun. There will be on Tuesday night a social event, being the event online this year. The social event will be an epic game Tuesday night with a musical overture, board games and card games. And some card games that we will propose are parts of the Italian tradition. So if you're not Italian, don't be afraid, ask the locals. And it's a mess of the beginning. The rules could be a bit difficult to learn, but it's also a lot of fun to participate and to join an Italian card game. You can also, if you want, learn some bad words, but please remember the code of conduct. Talking about social, and that's final. These are the links of the social means that we can use to talk to each other: the Mattermost channel, the Telegram group, and the Twitter account that we shall use for such events. And that's all, folks. Thanks, everybody. Let's move on. New people to admit. New people in the Telegram group. Well, fantastic. This was very, very effective.
Thank you, Davide. Maybe people have some questions to ask you about the BrainHack way to work. Probably this is the time to get questions from them. If there are questions. Somebody is asking, can you send the link of the Telegram group here? The PNC link does not work for me. And the link is there, kindly provided by Davide.
Okay. If in case you haven't noticed, the link page, the private one, password protected, that I assume everybody here has seen, will be updated. So at the very top of the page, you will see the current link for the current session. So we will have a morning session and an afternoon session for each of the 3 days. And the social event will have its own Zoom link. So we shall repeat this. Please keep on updating and keep on watching on the link page. Thank you.
Somebody is asking, can we choose the project we want to take part in?
Of course you can. There will be a time right after the first two talks in a schedule of the project pitches, in which each one of the project proponents will explain the project. And so you can watch the presentation and choose your better fitting project.
Emma is also saying that she will be doing a mini GitHub tutorial in her talk, which is appreciated.
Okay, let's give some statistics about the participants. We have 122 registered participants, which is a lot. Thank you very much for the enthusiastic participation. 66 of these are Italians, 34 from the rest of the Europe as a continent, not as European Union, 19 from Asia, 1 from Africa, 2 from North America, 4 from South America, and 0 from Antarctica. Amongst the participants, we have 46 students, 32 PhD students, 22 postdocs, 5 research assistants, 7 professors, and 10 have registered as others less freelance workers. So there's really a lot of people. Thank you very much for registering here.
Davide, there are a couple of important questions in the chat. One is Cristina Scarpazza, who is asking, if we decide to publish on BrainHack proceeding, will this prevent a further publication on a higher impact journal? This is a delicate question, of course. And I don't know personally. Do you know, Davide?
I'm not really sure of the answer, but it depends from the higher impact journal policies. So if they consider such publication as a per se publication or a sort of a preprint, so it can depend on the journal itself.
Yeah, it also depends on these new journals' status. Is this a preprint repository, or is this a real journal that aims to get indexed in the main, engines, search engines like Scopus, like all the rest? And yeah, that's. Is it peer reviewed? It is, right?
It is.
Okay.
So probably yes.
So my guess would be that we could try to publish something if it is a kind of preliminary pilot study, for which you might want to maybe extend the work for a future bigger project. This would be safe and wise, but unless we get more information, I wouldn't have an answer. Yeah.
Okay, I will simply ask the BrainHack community. So you will get an answer later in this day for sure.
Okay.
We will discuss
Thank you,
it again. You're welcome, Christina.
There is another question. Do we choose a project for every day or for every session or one for the whole event?
Okay, we encourage choosing a project and participate in it for the whole event. But if you choose an event, project and you feel like you don't really fit, you're welcome to try something else. But we really encourage you to stay with your project because you will enjoy it more.
Yes, please avoid to jump from one project to another because I think it is not worthwhile to jump. Davide, your audio isn't enough.
Yeah, I was just checking the YouTube comments. So if people are not in the Zoom session, can comment and ask for things in YouTube too. And there is no commenter in this moment, so it's fine.
Marc, est-ce que tu ferais pas un café?
Cristina, did you solve the problem with the two people joining your group?
Yeah, I think so. Enrico is already here, and Massimo will be here tomorrow.
Okay, great.
Thank you.
No problem. So we have 8 more minutes before we start with the Ignite talks, if you have other questions.
Nicole asked, will the project pitches be recorded? Yes. Every Ignite talk and the progress report is recorded, unless you asked us not to record your. On the other hand, when you work on group, in the working group, it's not required to record the session. Before starting the first Ignite talk, I have to send you the warm greetings by the other organizer of the BrainHack, Patrizia Bisiacchi, who is not here because he has to attend to all the lesson, but she will join us later in the afternoon. So.
Daniel told me he's ready, just a couple of more minutes just to stay on schedule. And in the meantime, I will ask him for permissions in private chat.
Okay, so we are ready. I'm happy to introduce the first speaker of the day and of this BrainHack, Daniel Merguez. He's a tenured researcher at CNRS in Paris, and he's interested in the organization of large-scale brain networks, and in particular in the emergence of network topography and in the relationship to cortical structure. So today, Daniel, are you here? Can you join? Can you share the screen?
Hi.
Hi.
Oh, yep. Hi.
Okay.
Oh, can you give me permission to share the screen?
Yeah. So
I
today.
think it's already given. Can you try?
I'll try. Yep, there it is.
Perfect. So today we talk about exploring cortical gradients across individuals. Gradients is like a trending topics in this last year. So I'm really happy and curious about his presentation. So thank you for joining us. And it's your virtual stage.
Fantastic. Thank you. First of all, thank you so much for the invitation. Thank you for organizing. This has been a weird and wonderful year in all sorts of ways, and being able to wrap it up with everybody coming together around some brain hacks is both wonderful, but also I think it's exciting to see how the community has been able to shift to accommodate and really thrive in many ways in the virtual space. So awesome, and really looking forward to seeing what's going to happen at this brain hack. And I should mention the work that led to characterizing cortical gradients in many ways really emerged through the BrainHack community and the interactions I was able to have with researchers around the world at BrainHacks and be able to develop the methods to approach these questions. And what I'm going to be talking about today in the context of this Ignite talk is some of the current trends in the field and some of the current challenges that we're facing, some of the advances as well as some of the open questions. So if I'll be going through some of the content rather quickly, of course, feel free to jump in and ask questions, but I'm going to try and keep it broad strokes, again, just in the spirit of a kind of brief introduction to some of the topics and things that might be possible to also explore and discuss during the next couple of days in projects. And specific to this theme of exploring interindividual differences, diversity, etc., I wanted to look at what was happening in the field with trying to characterize individual phenotypes using these kinds of gradient-based approaches. But to start off, I'd like to give kind of a bigger picture of what one of the uses is, one of the utilities of this approach, of this analytic approach. And I'm a big fan of going back to like moments in the history of science that were interesting and innovative and provided a new perspective. And this is no exception. If you think back like to Linnaeus and biology and the question essentially of how do we characterize different species and how they relate to one another. Historically, one of the key ways to approach that question was to think of it and to explore it in terms of a hierarchy of species in which there are different features that relate them to one another. And one of the quite unique innovations in ways of approaching and thinking about the relationship of different organisms to one another and how do we characterize that was by Alexander von Humboldt, who in his explorations and his, let's say, his travels came to an understanding of the way in which, in this example, different flora can be characterized based not only on the features of that species, but where they're located in the context, in this case, of altitude, as well as where they're located in terms of different climates, whether tropical or temperate climates, as you can see in the illustration here. What's really beautiful about this way of picturing it was that it situates individual features of these plants, in this case, within a kind of global context, within the space of how they relate to one another. And that reveals certain aspects that wouldn't be able to be characterized by looking at any one specific species alone. It helps to inform our understanding of the individual species within this grander interrelationship between them. The reason I'm a fan of this is that I think it raises one of the interesting questions that we can be exploring in brain research, in contemporary brain research, which is that we're very used to looking at individual regions and to try to think about questions of what they do functionally, of what their different features are. But of course, the brain is highly interconnected. And although we can draw distinctions between different parts of the brain, they're all just a few synapses. Any 2 points in the cortex especially are only a few synaptic steps away from one another. And so how can we also think about how these different systems relate to each other in the global structure of the whole? And that, in a kind of broad strokes introduction, is what these class of methods allow us to do and allow us to characterize. It's not about distinguishing with hard lines one region from another. It's about looking at how these different features relate to one another. And this is just a kind of summary figure of one of the key findings from some work from my lab about 5 years ago, characterizing the 2 dimensions of what could be termed connectivity space, to use the term from Mars and Javabdi. And what you see on the left is the first 2 gradients, which are, just to simplify, they're essentially like principal components. We'll get back to that. It's using a slightly different method. For now, doesn't really matter so much. But what it's characterizing is the dimensions that capture the maximum variance in connectivity. And what it reveals, just looking at the first 2 dimensions of that connectivity space, is a pattern in which you have this segregation between different sensory motor systems or input-output modalities, and then integration towards this core set of regions that can be characterized in the human brain as the default mode network, those regions in red that you see there at the kind of tip of the triangle, in which there's transmodal kind of sharing of function across different modalities that spans different modalities, integration across those systems. And this is consistent with what had been theorized previously based on macaque-monkey track tracing work by Marcel Messelin, which is shown on the right. I'm not going to go in detail into the methods today, again, just in the sort of context of this talk, keeping it brief, but I do refer you, there's some very nice methods work out of an introductory reading that you can do if you want to get into this. from Boris Bernhardt's lab is in the first link on the bottom left. There's also a really nice recent review article that introduces some of these methods very nicely by Claude Bahada and colleagues, as well as a link to some of the methodological approaches from Kuhn Hack that I do recommend checking out if you want to get more into some of the methods that have been developed to be able to do these kinds of analyses. But essentially what they reveal is a pattern in which we can talk about not only the distinct networks or distinct large-scale functional systems, but how the systems relate to one another. We can situate them within a kind of global hierarchy of different functional systems in which we have, as I described before, this externally oriented input-output systems at one end, which are segregated from each other, and they integrate towards this core set of regions of association cortex, most characterized there by the deep follow network at the center of that hierarchy. It helps to put these distinct functional systems into context, right? And to look at them as whole within a global whole, like that sort of mountain that, to go back to that initial metaphor from Humboldt's work. But of course, one of the challenges then becomes, how do we, what are the features that we'll use from the space to characterize individual differences? How can we explore the way in which these different connectivity spaces relate to differences across individuals and even differences within individuals from moment to moment? And just to give one kind of, again, broad illustration of where this is potentially a useful technique or a useful methodological framework, this is a really nice illustration from a review a couple years back by Rohir Mars, Passingham and Jababdi, where they are illustrating the value of projecting into a functional connectivity space for distinguishing different task-induced states. Okay, so to put that in other words, if you look at the box on the left, what it's showing is the representation of these two different contrasts. It's language and math from the Human Connectome project that you see in the center. And on the left, it's being shown purely based on anatomical space. So the way in which those different points are represented in caudal rostral and ventral dorsal dimensions, right? And they're quite intermixed because these patterns themselves, as you can see in the middle, are intermixed. There's different colored patterns that are interdigitated in various ways. As soon as you define a space based on the first two dimensions of kind of decomposition of functional connectivity data, it beautifully distinguishes those two spaces. So you can really just with a ruler draw the line between those darker dots and the red dots there. And what this helps to illustrate is that if we take the brain data, in this case, the resting state functional connectivity data on its own terms and use that to describe a low dimensional projection of that data, it enables us to characterize differences in functional states in a much more intuitive way. It's a coordinate system that's based on brain activity rather than based on almost the, let's say, the arbitrariness of the anatomical layout of these systems. And to give one brief illustration of where this is used, building on the prior example, here we're projecting 100 individuals into a three-dimensional space based on the different states during a working memory task. So it's, what is it, 0, 1, and 2 back, or sorry, fixation 0 and 2 in this case, from the Human Connectome project. And if we just look at gradient 1 and 3, so the top right of those three different plots, you can see that it really nicely distinguishes these three different conditions from one another. Again, with a ruler, we can get very high accuracy between the red and the blue dots, which characterize these different states on an individual level. But then taking that even a step further, the question becomes, to what degree do the positions within that space characterize a meaningful aspect of individual behavior? And what we did find was that performance in a 2 and a 0 back condition on the individual level was predicted by the position within gradient 3, that third gradient, which seems to distinguish these kind of task positive systems or multiple demand network from a kind of default mode network. That's the dimension that's being characterized there. And again, it just begins to raise these questions of, this seems to be meaningful, the way in which this space is occupied by an individual brain. What are the most appropriate ways to characterize that, and what are the methods we can use to do so? So there's been a lot of work in recent years, really couple of years, taking these basic approaches of using a dimensionality reduction tool to look at the first few dimensions of connectivity space, and then investigating how that varies across individuals. And the methods are still very much in development, I should say. So the tools are available, but then the question becomes, what are the features that are going to be most meaningful for capturing those differences on the group level? Is it the position of points? Is the geometry of that space relevant on the individual level? Is it looking at the entire spectrum as a whole? And these are the open questions in many ways. They're being explored in a lot of recent, very exciting publications. This is work by Hong and colleagues from Boris Bernhardt's lab, looking at autism spectrum disorders, and essentially, in brief, just characterizing a contraction of that principal gradient, that first gradient, from somatosensory motor to default mode, which can be interpreted as being a suppression of a large-scale hierarchy. So here, thinking about it in terms of a functional hierarchy, and what happens if that space is not as extruded or expanded, if those points are closer together and aren't as distinguished from one another. There's recent work that's been put up on bioRxiv by Gearin and colleagues, from Nathan Spring's lab, which show a similar phenomenon of a contraction of that gradient in an LSD-induced state across individuals. And in this case, also along the second gradient, showing that contraction from the visual cortex in the blue arrow that you can see there. And I've also done some preliminary analyses looking at a small group of sighted and congenitally blind individuals, and found a similar phenomena of a contraction along the principal gradient, so that in the congenitally blind group, the distance between the sensory motor areas and default mode is not as far. And it raises these questions again about, how do we interpret shifts in these individual points within this space? Is it specific to that dimension? Is it a result of what's occurring elsewhere along the spectrum that's being characterized by these dimensionality reduction techniques? And again, these are all open questions that are going to require further methodological exploration and development. But there seems to be meaningful differences that are characterized by these approaches. I also just wanted to briefly show, this is really beautiful work, taking that question of what features can we use from these basic analyses to characterize differences. And this is work by Richard Bethlehem, looking at age-related shifts across the lifespan, across a group of individuals. And just in brief, he's using, in this case, a measure of dispersion within networks across this gradient space to characterize differences both across the lifespan as well as in relation to general intelligence that's shown here. And it's a really elegant paper that just came out that I recommend taking a look at for creative and novel ways of really getting into the data and trying to understand, how can we best characterize what's occurring in this, no doubt, low dimensional space representation, but certainly more complex, because it's no longer reducing the data to a small set of networks that we can just talk about in isolation from one another. It's trying to look at how these networks and how these systems relate to one another. So certainly, as I've mentioned a few times now, the methods are challenging here. This is a new approach. It's not taking each point in the brain in isolation, but looking at how they relate to one another over space. And that requires novel methods development and looking into other fields and asking new types of questions of not how do we look at individual values in the brain, but how do we look at the spatial relationships between them. And one of the groups that's been doing very cool work, innovating methodologically, has been Koen Haak and Christian Beckman, who've been developing a class of methods to model the spatial statistics, to look at how these trends over space vary differently across individuals. And I won't go too much into the methods here, but what's quite, I think, interesting and also relevant in terms of the BrainHack context is what's super cool about the approaches that they've developed is that in asking these questions about how can we develop methods to look across space, they actually went to statistical techniques that were predominantly developed in the oil industry to try and predict the location for drilling. And the reason I mention that is that this is exactly where the kinds of spaces that happen during BrainHack are really exciting, because there are certainly fields that are asking and have been asking for many decades similar questions to the ones that we may be asking now. And ideally, BrainHack does provide a kind of space where people can come together and think creatively and try out new things that wouldn't necessarily be at the core of their daily research interests and approaches. And so this is one example of where thinking about questions that would be coming from a completely different field in a different way allow for exciting innovation to take place. And so what they've been doing, in addition to developing these basic methods for looking and characterizing the statistics over space, is to take both univariate and multivariate approaches to looking at how the low dimensional representation of connectivity can predict behavior. And they've also developed some very exciting multivariate approaches for doing that using canonical correlation analysis, as shown on the bottom right. So, like, one of the key questions when starting off with any new method, and again, this is where it's a good lesson in really how to approach this methodically, is to assess the reliability and compare these methods with one another, and also to assess their accuracy for predicting individual differences. And in a recent really comprehensive paper by Hong and colleagues from Mike Millam's lab at Child Mind Institute, looked at 3 different classes of techniques, Laplacian eigenmaps, diffusion map embedding, and principal component analysis, and compared their reliability as well as their ability to predict individual differences. And I won't go again into the details of this paper, just in the interest of like keeping this kind of broad strokes. But one of the exciting points that they found, that I find personally exciting, is that principal component analysis, a method that we're probably all familiar with, or is really the most approachable of all of these different techniques, seem to be the best in terms of reliability. And it's good to know that. I've been using, let's say, slightly more opaque methods before finding this out, and this has been a relief to know that really straightforward approaches that we can have a more intuitive understanding of what it's doing to the data are the most reliable and effective. And it's been valuable to have this additional insight that's derived from this paper. But bottom line essentially is that these techniques do appear to be largely both reliable on an intra-individual level, as well as accurate in terms of being able to predict variance across individuals in different behavioral measures. So to kind of wrap things up with the final thought about other kind of approaches or questions, or is that all of the content that I just showed you, all of the different examples, was focusing on connectivity space. It was focusing on these low dimensional representations of connectivity and how we can use that to characterize differences across individuals or describe brain organization. But one of the findings, and what actually got me going in this line of work, was this observation that's illustrated in the figure on the screen here, that shows the spatial relationship between these different systems. And essentially, like, to summarize this, the distance along the cortical surface, just taking out a tape measure and measuring distances along the cortical surface, the distance along the cortical surface helps to understand the spatial location of regions of the default mode network. So frame that another way. The points that you see with the white circles inside the red things, that's the default mode network, and it's located spatially equidistant from primary sensory motor areas. There seems to be a very tight mapping between the space. In other words, the geometry of the cortex is predictive of the location of these large-scale functional systems. And for me, that's super cool as a kind of way of understanding the way in which these large-scale systems relate to one another and what might be accounting for the roles that they take, the functional roles, kind of hierarchical relationship, and potentially the, let's say, an anatomical scaffolding for an underlying model of cortical dynamics and how information would be transferred over the cortical space. Given this observation, I think one of the places where there's a lot of space for exciting new development would be to look at how variance in specifically these distances, this intrinsic geometry over the cortical surface, how that might vary in a meaningful way across individuals. And there certainly has been enormous work looking into various features of cortical structures, such as thickness, probably has had the most attention, but also looking at surface area in a local level. Which has had, let's say, limited applicability. But one of the spaces that I'm excited to explore is the way in which specifically these axes of organization, from primary regions to default mode network, in terms of space, might also be relevant in thinking about questions of interindividual difference in cognitive function. So I did want to mention that. And just to kind of summarize, yep, we're good on time. So yeah, it appears that these gradients are able to capture a functionally meaningful arrangement of systems, and we can characterize differences across individuals in ways that are reproducible, that are reliable, and that in many ways are accurate. But I think one of the key places that requires a lot of innovation and deeper understanding is, how do we interpret this? What are the features we should be characterizing? Is it appropriate using these specific class of techniques to talk about differences in values between individual points, or do we need to look at them as whole spectrums? And I think that one of the ways in which innovation is going to be able to happen here is through looking at fields outside of our own to how other folks are dealing with these questions of network analysis, dimensionality reduction, and characterizing and comparing these spaces with one another. So with that, I want to, of course, thank my funders and lab in Leipzig and wonderful collaborators. All the code that's been involved in the various analyses can be found on my website from the lab there, and the data for the work that I showed from my lab was from the Human Connectome project. I should also, that's there, yeah. There's a special issue that is just being wrapped up, but most of the articles are available now with NeuroImage. Everything's openly available there on gradients and brain organization, and there's some very exciting recent work that I recommend checking out. So looking forward to discussing these topics over the next few days, and thank you again to the organizers for the chance to chat a bit. Yeah, wrap it up there.
Thank you, Daniel. Are there any questions you can ask personally, or chatting, or on YouTube you can send comments, so we are open to different modality.
I'll be around, so I know it's an intimidating space to. Oh, go ahead though. Yeah, sure.
I have a comment. I saw in several of your graphs the triangles, and there is the part optimality theory, according to which the evolution led to some constraints, biological constraints, but led to trade-offs. And these trade-offs have this configuration. So the configuration of a triangle. In which sense? In the sense that decreasing a particular feature led to an increase of another feature. I don't know if I explain well. So the basic idea is that these triangles reflect evolutionary trade-offs. So a way to cope with evolution leading to a particular balance between particular features. And for example, in your first picture, I would think about the full mode network in one extreme and the sensory cortices in the other extreme. And I would thought that, yes, there might be a trade-off between integration and segregation, between the defining features of these networks. So would you think that this idea of trade-offs, evolutionary trade-off, might fit your data, might be of interest in interpreting your data? I'm just wondering.
Yeah. I mean, I'd be interested to understand more about some of the specific examples where where this has been observed in terms of that triangle structure and what might lead to that. It'd be super interesting to find out and discuss that further. I should just mention that there has been quite a bit of work recently looking at cross-species relationships. Ting Hu has an article out actually in the special issue comparing macaques and humans, and finds that precisely there's that extension of the tip of that triangle as you look across species. So really specifically addressing that question of evolutionary change, as well as looking in rodents. So I think there's a lot of space there for comparing and thinking about that. But I'd be curious more also why this kind of triangle shape is relevant to what you're describing in terms of evolutionary trade-offs. So maybe it'd be fun to discuss that over what the examples are.
Yeah.
Sorry.
There is another question in the chat.
My computer's a little bit slow.
There is a question.
Yeah.
There is a question.
Please.
Question is
my computer is
no problem.
acting up.
Is provocative. Do you think that this might be an interesting future perspective to have a more no, sorry, this is the second question. First one was, can gradients be considered as a rebranding of SVD? What is your opinion on that? Singular value.
Absolutely, it's totally, it's like 100%, absolutely. And I do appreciate I appreciate the tone of the question. Totally. And when I presented this work to more let's say like biomedical engineering, mathematics oriented audiences, and they're of course like, you can't use the word gradient. What are you doing with gradient? Has a meaning for us. And it's totally rebranding. Yeah, this stuff has been done in the I mean, Friston, like everything, Friston did it in the 90s, right? Doing PCA on functional MRI data has been around for a very long time. Why haven't we really widely adopted it? You know, and I think part of it has been a branding issue. I guess there are two sides to that. So I'm going to give you the really critical side, which would be, yes, it's a rebranding. In the way that resting state networks were rebranding of ICA, independent component analysis components, right? ICA components. It provides a linguistic framework to enable a field that isn't familiar with the techniques to be able to talk about and interpret those methodologies and the output of those methodologies. So it certainly was that. Now, the other side of that is that, for, I guess, personally, what's really underlying the use of these techniques are these observations regarding the geometry of the cortex. And there, I think it's fair when we're talking about changes in features over space to use a term like gradients to describe those. So it's not purely a rebranding, it's an accurate characterization of how a feature space relates to, in this case, the physical space. And because that was always really the focus for me, trying to understand these spatial relationships and progressions, and the class of techniques for dimensionality reduction was in many ways secondary, that also was a motivating factor behind using the term in that case. But I think, it's a rebranding, but one that shouldn't only be seen with skepticism, because it does enable for methods that are, let's say, unfamiliar to a field to become approachable. and that has a lot of value, I believe, as long as it's
Okay,
done.
for the sake of time,
Somebody's
last
conscious.
question. One
Oh,
last
sorry,
question
yeah.
is regards whether by gradients researchers usually refer to cortical gradients, or there is also an extension to subcortical structures. I guess the answer is yes, there is an extension. Yeah, because there are.
Yeah, the answer is yes. There's a lot of work. One really cool paper just came out in Nature Neuroscience by Ye Tian and Zaleski, and yeah, I'd refer you there. There's an atlas based on looking at these, in this case, gradients and connectivity of subcortical structures that might be interesting for you. Yeah.
Great, thank you.
Happy to discuss more. Thank you.
Merci, Daniel. Thank you very much. Now it's time for
sure.
the next speaker. Can you show up if you please?
Hello.
Hello, Emma.
Hello.
How are you doing?
Fine, thank you.
Okay. Emma is an independent postdoc researcher and working in a number of open science projects and accessibility to open science with the Turing Way, and she will explain what the Turing Way is. And she's developing open reference collection for British plant communities and a verification project in archaeobotany. So thank you very much, Emma, for being here.
Thank you.
If you want to.
Yeah, I'll just share my screen. I hope my computer is gonna be good today. So can you see something? Oh, just across. Now, can anyone see anything? I'm going to present.
Now,
we
yes.
do now.
Yeah, okay, I'm just going to present it to make it bigger. Sorry, my computer's quite slow when I've got Zoom on, so it should work in a second. Oh, is it working? Yes, it's doing something. I'm also going to turn my captioning on as well, so that people can see as well. Hopefully that will work. Okay, so thank you so much for inviting me to speak today. As David said, my name is Dr. Emma Caroon, and I work as an open scientist. And this is doing a number of different things. I do not study the brain, so I'm not going to talk about the brain at all, but I do study the decisions made by it. As David said, I am actually an archaeobotanist, so a type of archaeologist or archaeological scientist. So this means I study the actions of past humans and the decisions that they made in terms of the exploitation of the environment. I investigate ancient plant remains that are found in archaeological and paleoecological deposits. And I tried to find out what plants were used, how they were used, and how humans have impacted on past environments. But today I'm actually representing the Turing Way, which, if you don't know what this is, it's an open source, community-driven guide to reproducible, ethical, inclusive and collaborative data science. The project is led by Kirsty Whitaker at the Alan Turing Institute, and also the community manager is Malvika Sharan. So those are the people to really get in touch with if you want to get involved. And the goal of the Turing Way is to provide all of the information that data scientists in academia, industry, the government, or governments, and the third sector need at the start of their projects, to ensure that they are easy to reproduce and reuse at the end. So the book started off as a guide for reproducibility, covering version control, testing, and continuous integration. However, technical skills are just one aspect of making data science research open for all. So this year, and starting in February 2020, the Turing Way expanded to a series of books covering reproducible research, project design, communication, collaboration, and ethical research. So this is now about to be expanded further again with work that has been done in the recent November Book Dash. And if you want to get involved in the project, we would love to have you come and be a contributor to the book. So here are all the links, and I'll put them up at the end of my talk as well, while we have questions. And we have a newsletter that goes out monthly and lots of different events that happen regularly. So please do get in touch if you're interested. So my talk is about reproducibility and collaborative working. So I'm going to start off by talking about what reproducibility or reproducible research is. Then I'm going to go on to talk about why, and probably hopefully convince you why you should be doing it. And then at the end, I'm going to talk about how you actually do reproducible research, and specifically how you can work collaboratively using version control systems. And at that point, I will do a bit of a mini tutorial on GitHub and a bit of a show and tell, I suppose, of different project repositories to show you what can actually be done with it in larger projects. I think that's quite a good way of looking and learning about GitHub. So what is reproducibility? So there are definitions, different definitions of reproducibility, and these change really depending on your discipline. So in terms of the Turing way, we actually define and make the distinction between reproducibility and replicability in terms of the data. So a result that is reproducible can be defined as when the same analysis steps are performed on the same data set consistently, and it produces the same answer. Whereas a result is replicable when the same analysis is performed on different data sets and produces qualitatively similar answers. But you can actually go further with this, and in terms of reproducible research as an approach, because really we want to build methods that are robust. So a result that is robust when the same data set is subjected to different analysis workflows to answer the same research questions, such as using one pipeline that's written in R and another written in Python. And then we would be getting a qualitatively similar or identical answer being produced. And that means that we have these reproducible results that show that the work is not dependent on one specific type of analysis that is being performed. So it actually gives you the answer with different forms of analysis. So we know it's a robust bit of research. And then you can go further and make it generalizable. So generalizable goes further still to combine replicable and robust findings. And this is an important step towards understanding that the results are not dependent on a particular data set, nor even a particular version of analysis pipeline. So you're looking at incorporating more data sets and also more forms of analysis to find the same or similar answers qualitatively. Sounds like quite a big job really, and it actually is, but I think it's all about planning, which I'll talk about a bit later. So why might you want to actually do reproducible research? So there's actually many benefits to you as a researcher and to the wider scientific community, and also I think the public as well. So the first thing is about tracking the history of your research. So this includes the data, tools, methods, code, the documentation used in your research. So this ensures research sustainability, which is something that's talked about quite a lot at the moment. There also gives fair acknowledgement to the contributors on your project, so your team or the wider community, and also it maximises the usefulness of your research to others. So for me, it's really about keeping thorough records of what I'm doing to help me actually remember where to start if I've had a bit of a break from a certain research activity. So that's quite a personal thing, but I find that really works well for me. So secondly, to facilitate collaboration and the review process. So reproducible workflows actually enable other researchers and also reviewers to have full access to all of the different parts of your project that are necessary for validation of the research outcomes. So this means that you will publish validated research and avoid misinformation. Thirdly, it helps you to write papers and other research outputs efficiently. So documenting the project throughout will make writing up easier, and you're likely to already meet the highest level journal guidelines for the availability of data sets and metadata. Fourthly, to credit work fairly. So other researchers can use and reproduce different parts of your project, such as the data, the code, and the protocols. So all of these different parts you can give digital object identifiers, so DOIs to, and therefore they are all individually citable pieces of work. So this means that you will get fair credit for this work, and your research is likely to have more impact, as you will be cited more for those different parts of your research outputs. And actually, it's recently been found that articles that include statements that link their data have 25% higher citation impact on average. So that's something to really think about. And then lastly, ensuring continuity of the work. It's kind of similar to my first point, but actually the progress of reproducible projects can be easily tracked and continued, allowing you and others to build on your research. And actually this again is something that makes research much more sustainable, where we can have different people working on teams, and they can take over very easily from projects from another member of the team. So how can you actually do reproducible research? So there isn't just one way of doing reproducible research. There are many different ways of doing it, and there are lots of different software and tools that you can actually use. So things that I think you need to consider if you're thinking about planning a new project to be reproducible is your own skill level and your team's skill level, because actually working with an open science approach does actually involve a new skill set, and that's something that you need to consider when you're planning it. I am not from a computer engineering background. I'm a research scientist by background, so I tend to go for a more simple way of working reproducibly, although I use some advanced version control systems such as GitHub, which I've had to upskill myself in. So it's something that you need to consider when you want to do reproducible research, but you can do it in a very simple way, which I'll explain in a minute. Your discipline, you also need to consider your discipline skill level. So my particular discipline has quite a low skill level generally, so in terms of open science skills. So I actually have to consider that, because I want them to be able to access my work and use my data. So I want to make sure that it's at a level where there's no barrier in terms of a skill level for them to access it. And then lastly, accessibility to tools and software. So if you want to work openly and reproducibly, that means then accessibility to your work is really a key consideration. So lowering this barrier in terms of skill level, but also accessibility, means that more people are going to read your work and are going to use your work. So try to use open source and free to use software and tools where possible, so that these barriers are lowered. So other things to consider when starting reproducible research projects is really you're looking for transparency and collaboration. So in terms of transparency, you want all your work to be as open as possible. So this could be from the outset of your project, or just at the end of your project, maybe in a research compendium, which I'm going to talk about in a minute. But it does depend on the sensitivity of your data. I don't really have issues with sensitivity, because everything I study, well, it's all dead. It's been in the ground usually for a long time, and I don't study human skeletons. So all of the things I study are plants. So there isn't really that human sensitivity there, but I know probably in your field with the brain, there is some issues with sensitivity. So that's something you need to consider, is when can your data actually become open? How can it become open? Can you make it anonymous so that that kind of sensitivity of the data is taken away and it can become open for other people? But also collaboration is a really important thing to consider. So this year particularly, we've all been working very remotely, collaboratively. So it's about logistics of that, and also how are you going to record that collaboration? And this is where you need to involve version control. And I will talk about that in more detail in a second. So if I come back to my first point there about a research compendia. So the final output of a research project is still a published research article. And actually to make your work into, or that article into, a reproducible piece of research, it needs to have with it all of the information about the project attached to it, so that it is transparent and therefore reproducible. So you can do this by using a research compendium, which is a good way to structure this information into a set of files. In these files, there must be all of the information needed for somebody else to reproduce your work. So if you have a look at this basic one here in the picture, it includes the descriptions of methods, raw data files, analysis of your data and codes, and also a README file, which is like a landing page for your project and sort of introduces what it's all about. And these files, they can actually be published as a supplementary file set with your article, although sometimes if your article is not open access, it also means that the supplementary files are not open access. So if you want your work to be reproduced, it really needs to be put in an open repository. So you can actually use research archiving sites such as Open Science Framework or Zenodo to put the set of files in there, or you can use a versioning platform such as GitHub or GitLab. You can actually also package your research compendium into an executable research compendium. So this means another researcher can easily rerun your analysis sort of automatically. I'm not really going to talk about that because I suppose it's out of my skill set, I suppose. I'm much more in the simple way of doing things. Oh, there's my picture. There we go. So in terms of version control, version control really it's an approach to record changes in a file or a set of files over time so that you and your collaborators can track their history and review the changes. So the benefits to this are that you can actually go back to previous versions of your work. So maybe you've done a whole morning's work and you just want to scrap that, so you can step back really easily. It stores a history of all of the changes that you have done and when you've done them. You can work collaboratively on documents, so seeing what other people have done, and then you can record those contributions of the team, so therefore allowing fair credit for the work done by different members of the team. So actually, how can you do this versioning control? So the most simple way of doing it is actually just to you rename or have a versioning way of renaming file names. So that's a very easy way of doing it. But then you can actually start to use tools. So you can use simple tools like Google Drive or Dropbox, and they actually record for you different versions of your work that you can go back and go back to previous versions. Or you can start to use more advanced tools such as GitHub or Subversion. So what are the difference between these simple versus these advanced version control systems? So really, it's about how you're working with it. So with a simple version control system like Google Drive or Dropbox, they just record different versions of the file so that you're able to step back to earlier versions, and it's kind of a linear way of working. Whereas with advanced version control, it allows different team members to make branches along the main file. So in this diagram at the bottom, you've got the main file or the main branch of your work, but actually each team member can put their own branch off that and make changes to the work. Other people can then review those changes and they can be merged back into the main of documentation or code or whatever part of the project you're actually working on. So it's kind of a much more collaborative way of working that records everything as you go along and helps you to merge those changes easily, sometimes not easily, but easily into the project in most cases. So what actually is GitHub? I'm just going to check the time. Yeah, I'm going to speak a bit more quickly. So what actually is GitHub? So GitHub is this amazing research tool that is free to use. It's not just for computer engineers, although I think it's predominantly still used by computer engineers, but it's also really useful for research scientists or anyone working on collaborative projects. So it uses Git, which is the open source version control tool. And GitHub is actually the online interface that uses Git. And as I said, it's free and open to use for anybody. So why is it actually really useful for reproducible projects? So the great thing about it is that you can store all your project documentation, data, code, in one place called a repository, and you can create web pages of this repository and archive it onto Zenodo, which is a very long-term open storage repository controlled by CERN. It's actually also a web interface for version control. You can also work collaboratively by using the project boards and issues and pull requests, which I'm going to explain in a minute. So how can you, hello? Research scientists or anyone working. Oh, someone was talking. So yeah, if you've got a question, put it in the chat. I can't actually see my chat at the moment, so I'll answer the question at the end. So how can you start to use GitHub? So there's a number of different ways. You can actually use it directly in your web browser, and that's the easiest way to use it. That's the way I use it, and you can use it completely through the web browser. You don't have to use it in any other way. You can download the GitHub desktop app to use it more locally on your computer, but then you have to push all your work up onto the GitHub. So I suppose it's the repository. You can also use it via the command line using Git. So any app that does that you can use, or if you use RStudio, you can actually you can link that to your GitHub account, and it actually pushes all your work up onto a GitHub repository automatically if you set it up. So probably a lot of people are using RStudio. So how do you actually get started with GitHub? Well, you just sign up for free. So this is the login page. We at the Turing Way have just been working on some new chapters in the collaboration guide. So if you're really new to it, you can have a look at the Turing Way, and there is a guide to getting started with GitHub. And it has step-by-step instructions and tips and resources for you to really get started. So I would suggest having a look at that. But the things that I found quite hard when I started using GitHub was that it's kind of like another language. It's a new skill that you have to learn. So it does appear quite daunting to new users, but actually I haven't actually found it any more difficult than learning a new bit of software like, I don't know, Word or, you know, a Microsoft Office or Microsoft Teams or something like that. It's just, you have to just get used to all the different buttons and all the different words which kind of go with it. So the two main things that you have to grasp are, the first one is Markdown, which this is how you write all of your documentation within GitHub, so that it can be rendered as a web-ready version of what you've written. And it's a very simple text to HTML conversion tool for web writers. So what you basically do, if you have a look on the right-hand side, I'm going to use my thing, you just use a few symbols to actually change the formatting of your text. So you will actually see your text within the document, and you're just adding the symbols to tell, basically tell the page how you want it to actually look when it's rendered into a web page or on the GitHub repository. So it changes. If you look here, you've got header 1, you're adding a hash symbol, and it changes it into the largest heading. So it's actually a really easy thing to learn, and there's lots of cheap sheets out there that you can download and just use. Really would only take you half an hour to kind of learn how to do it. The second thing you have to sort of learn is the specific GitHub word. So I've talked about a repository, or a repo is what it's often called. That is your project page where you put all of your files. It has a main branch, which is called main. Sometimes it's called master, but they've actually got rid of that term because it's not a very good term to use. So it's now called main, and so that's the main version of your project. And then you can branch this off into different versions that different people are working on, that will then be merged by somebody back into the main branch of your project. The other thing to get used to is the commit button. So this is basically the save button. You can add comments before you commit changes. And then there's these other things, issue, which are all the things that you need to do in your project that you write down as an issue. And then a pull request is actually when you finished some work on your branch and you want it to go back into the main repository, and you actually do a request, which is like a form to tell people what you've done, and then it can be merged back into the branch. And those main branch, those are the main words that you need to learn about it. So what I'm going to try and do now is I'm going to try and do my bit of show and tell. So I'm going to just come off my presentation and go on to GitHub. So first of all, I thought I would show you. David, how much time do I have left?
I would say 5 minutes about. But
okay, that should be fine. That should be fine. Thank you. So when you've got a GitHub account, you log in and you'll see a page like this. This is like your home page. So mine has got repositories on this side. It's also got recent activity that's been going on basically on my my GitHub account. If you want to start, what you want to do is you actually want to create a repository for your project. So you can go over here, there's a really nice green button that says new, or you can go on the black banner and you can press the plus. What I've found with GitHub is actually there are more than one button to use for each actually thing that you want to do. So you just have to find which is the best way for you to do everything. So if you press the plus button here, you don't actually want new project. What you want is new repository. So you just click on that. And then this hopefully my computer, there we go. So it brings you to this form. So this is creating your project page. So I'm the owner of it. The repository is, I'm just going to put test. Ooh, I'm going to give it a name, test 1. That's available. You want it to be public because it's going to be an open project. You can make it private if you want, that's okay. But remember, GitHub and open working is about making it open so it's reproducible. You're going to add this README file, which is your landing page file. And I'm going to talk about licenses in a minute. So then I'm going to click this green button, create repository, and it's going to bring me to the basic repository page, which is like the page that David showed you that he had set up for the BrainHack. Hopefully it's going to work. Right, so this is the page that you get. So you see you've got, this is your project page. We're going to add everything on your project. So here on this blue sort of banner here, these are the files that you have in your repository at the moment. So at the moment I only have a README file. And it has the .md. That means that it's a markdown file. So you're going to have to write that in markdown because it's one that's going to be rendered. So this is where you're going to want to, so here is actually the file here. This is where you're going to want to edit that to start off with, because that's going to be the page that everybody sees when they come into your project. And I'm going to show you some versions of this in a minute. But if you want to edit it, then you just press the little pencil and it takes you to the edit page for that particular file in your repository. So this is what it looks like. It looks a bit like a scary computer coding page, but don't be scared by that. Okay, so I can edit the file here. If I want to see what I've edited, then I can press this preview changes and it shows me what it's going to look like when it's rendered. So that's what it looks like. If I want to change it, maybe I want to add like, I'm going to do my 2 dashes for the second heading. What is about? Oh, now I can't type. And then that's going to look second heading, something like that. So you're going to add in there. What I suggest you actually do, so once you've put in and edited your README file, what you need to do then is commit the changes, which means you're going to save the changes. So a good practice with GitHub is to write a label for what you've done, what you're saving. So I've done edit the README. And then I'm going to do it. And if you look at the bottom, it says commit directly to the main branch. So that means it's going into the main branch. You could put it in a new branch if it's something you want to work on a side branch, but actually I just want to the main branch because I'm the owner. So I'm going to, so now it should come up on the, yeah, so I'm still here. So you can see now this is what I've added on the README file. Okay, so what you can do from here then is you can actually, I'm going to show you a more basic. So this is something I'm working on at the moment. This is a repository for my open science project. So these are all my files that I've got here in there. I have edited my README file as an introduction to my project. So I've got lots of different things here, what the project's about, who is involved, what things I need, because I'm trying to create a community project, how people get in touch, how people, more things to find out about, like the code of conduct. That's a good thing to have, contributors guidelines as well, and a bit of a glossary and a license. And a license is very important to put, if you're going to have your repository open from the beginning, you must put a license there to start off with, because a license allows people to reuse and rework and be able to really access your work in the way that you want. So, I mean, the most open license is a Creative Commons Zero license, which is recommended for data sets. But for most other bits of work, like a repository with documentation, it's better to have the CC by 4.0, which means that it's open as much as it can be to be reused, but people have to give you credit, is the basic idea behind that. And you can actually import these licenses from the Creative Commons website to be copied into a file. What I do is I tend to put these banners at the bottom of my README file, and then I put an actual license file within the document here, so that's the longer license. The other thing to take note of here is that I've made this into a web page, so you can actually really easily just in the settings at the top here, you can enable GitHub pages, which turns it into a basic page. But actually, it's very easy to then get a template which you can put into the repository and make it into a web page. So I'll just show you. So all of this here, anything that's got the .md is going to be a page on my web page. So I'll show you what that actually looks like when it renders. And you'll see what it lands on is my README file on the website. So this is exactly the same file that I've got on my GitHub README. So this is the file, same thing. And then all the other markdown files are these files at the side here. Okay. And then what I also wanted to show you is the Turing Way. So the Turing Way is a very large community project. My project has me as one collaborator by myself at the moment, because it's just starting. But actually here you can see in the Turing Way, there's 147 contributors to the project. Actual book, this is where it's stored in the GitHub repository, and then it has a Jupyter book, which makes it into the published online book. And everybody in the contributors, what we do is we use the issues and the pull request to actually make additions to the book. So if I want to start something new in the book, this is in the master, I would create myself a new branch, and then I would actually go for looking at creating an issue and working on something. So getting something, so this is one I've been working on. There's something wrong, some wrong images in the chapter that I'm working on. So I've told people about it because I need some help with it, some technical help. And then once I started that, then I can start working on it, where I start my own branch and I do a pull request to say, this is the work I'm doing, but it's work in progress. So this is how we do it. I'm trying to get this one up to show you. So when it's work in progress, I need someone to actually review the work, and then it will actually be merged into the actual main branch. But that probably won't happen for a while. So actually what happens is you fill in a form to say what you're going to do and you start working on it. So with GitHub, it actually records with the versioning system, it records what you're doing. So these are all the things that I've actually done. So I've been adding links in to the glossary to link words in the actual book to the glossary at the end. And this is all the work that happens. So it's all recorded here. So this is other work. And then the people who I'm working with, Paul, he's been doing other things. He leaves me some comments. Malvika is asking me how I'm getting on. Am I happy to merge it? And then we can leave comments. So it's a really collaborative way of working, that we can actually make comments and we can actually do our work where it's all recorded. It records everybody that does it. So it shows over here who are the contributors of this work, and that's all recorded in the repository. And once that's finished, somebody will approve it and it will be merged into the final book. And the branch then will disappear as a part of the main book. Okay, right, that's all I think I've got to talk about. Let me just go back to my Turing Way slide so you can all see. So thank you so much for listening. I hope it's been helpful, and I'm very happy to help people this week if during the brain hack if they want a bit of help with GitHub. So yeah, please have a look at the Turing Way, and especially the new chapter on getting started with GitHub, because there's lots of really great instructions there and resources that you can actually use. So thank you very much.
No, thank you very much, Emma. This has been a very comprehensive and clear tutorial / talk. end. If there are any questions, quick questions that anybody could raise, also on YouTube comment, please feel free.
Just want to thank Emma, because it was a very interesting introduction on GitHub. Thank you very much.
Thanks. I would really recommend looking at the Turing Way, because I think it's a very unique project where it's a very large community and it uses GitHub to the fullest extent that it can be used. So we're very happy to have people who know nothing about GitHub come to be contributors, because we take any contribution. If you want to help with the typos in the book, so you've seen some issues there, editing, you've got ideas for case studies that will go into the book or new chapters. It's really the smallest thing that you can do we're happy with. So and we're very happy to upskill you in working with GitHub, and particularly in the collaborative way. So yeah, and we have regular book dashes, which is like a whole week of intensive work. But we also have collaboration cafes and co-working sessions as well, so going on every week. So yeah, come and see us.
Thanks.
Thank you very, very much. I don't see any questions in the chat or in the YouTube. And I think we can go on with the project pitches as in a schedule. Thank you very much again, Emma. And see you soon. Are the proposers ready? I think I will call them in alphabetical order as in the website. So Mohammed, Benedetta, Andrea, and Valentina are the first proposers. Is anybody willing to talk about the projects?
Hello. I have a short presentation about the project. Can I share my screen?
Please do.
Mm-hmm. Okay. Can you see my screen?
We do.
Okay. Our project title is about finding the association.
I'm sorry to interrupt you. Feel free to show your face, so we'll be easier.
Oh, okay.
If you want, of course.
Okay. Can you see me? Yeah.
Yep. Welcome.
Okay. Our project title is about association, to finding the association between the microstructural white matter based on diffusion MRI and personality trace based on no FFI test and the Human Connectome project dataset. For the first step, we did diffusion MRI analysis based on preprocessed diffusion MRI data that provided by the Human Connectome project, and then use DSI Studio software to do automated atlas spectrography and derive 7 diffusion markers from 68 main fibers of the brain. And as I said, we use no FFI test for personality. Also, we remove the because we are going to explore the role of white matter. We removing the genetic similarity, and we remove twin sibling cases, and we are these pure cases. And we are, as I said, in the BrainHack, we are going to use a statistical approach regression and to build a regression model to investigate the association between the microstructural of white matter and no FFI test. if I these are private. Okay.
No, thank you very much, Mohammad. Going on the next proposed group, which is, if I do not go wrong, the Umberto group, Granziol.
Yes, can you hear me and see me?
We can hear you. I cannot see you at the moment.
Yes, I can see him.
Okay. So I will share my screen. Okay, I will try to be fast as my colleague. Okay, this is a network study. I will try to show you the main results and try to propose a project for the BrainHack working group of today. Basically, it is a network study. All of you now knows networks, and you, especially in psychological fields, but not only, you know that in the last decades, a lot of studies trying to link the human domains, cognition, personality, emotional function, etc., have been proposed. The problem is that none of the study, at least to our knowledge, mine and my supervisor, no study tried to collect them all together and tried to define a possible network structure of all the phenomena related to the same entity, the human brain. So, at least until now. This study tried, with the help of the Human Connectome Project database, the same of our colleague, to identify the main human domains, to try to define an architecture of such relationship and try to understand if such architecture can contemplate possible causal relations using innovative procedures like exploratory graph analysis and Bayesian networks. We will work with 1,206 participants, 656 female participants, with 38 measures, investigative, cognitive, mental health problem, behavioral problems, etc., and demographic variables. As I told you, we will use exploratory graph analysis and Bayesian networks, two very famous in the last 5 years, I guess, procedures, at least in psychology, of course, in using R programming language. I'm a very big fan of R, so we will use it. Using in our previous study, we reached this result. So 7 domains: mental health domain, substance use domain, externalizing problem domain, pain, delay discounting task, high cognitive function, and low cognitive functions domain. And with the classical approach of networks, so exploratory graph analysis, with very good results in terms of goodness of fit indexes and also stability of the network. We understood that some domains are related from a correlational point of view, but this point of view is good, but lacking of some information. That is, we do not know possible causal links. So we decided to use Bayesian networks, basically are networks, but the edges have a specific direction. So we used the same database. We trying to define the model passing from this network to this network. So we were able to understand that a lot of domains are related in using a causal pathways. So we reached the conclusion that we are ready to see the 7 domains with both correlation and the causal pathways, but we do not know what happened trying to run again the model on demographic variables such gender and also clinical versus non-clinical subsample. So in our working group, we will try to recreate the same models, both exploratory graph analysis and Bayesian model, but considering them even separately between male and females, but also for clinical and subclinical sample. I hope that was clear, and I thank you for your attention.
Thank you very much, Umberto. And now, in an incredible misplacement of alphabetical order, I apologize, my fault. Patrick, Frederick, if you want to introduce your project.
Sure, give me a second. So, can everybody see my screen?
We do.
Yes.
Perfect. So compared to the previous two really exciting projects, this one's a bit looser, and it's based on the idea that there may be different phenotypes in the brain anatomy based on observations of individual differences in the motor cortex or motor hand knob. So let's start at the very beginning. As you all know, in order to investigate different brains or groups of participants and their brains, we typically have various individuals which we register into a similar or common space, for instance, the M&I space. And this typically works well when we are looking at group differences and is a necessary step to make things comparable in order to do statistics. However, this comparability comes at a price, namely that the individual differences are not well preserved. This can be especially well seen when we're looking here at the hand knob in the standard space, which are painted in blue. So in a study by Kaulo et al. 2007, where they investigated the appearances of this hand knob area in 250 participants, they saw that in both males and females, this omega shape is quite prevalent, with up to 80% of participants showing either this shape in the left or right side. However, the whole scenery becomes a little bit more complicated because there are also different other shapes that obviously cannot be captured by something like a standard brain. And also these different shapes, like the medial asymmetric epsilon, the epsilon shape here, etc., differ between, in their case, males and females. So there may be some relevance for sex classification in this different morphological phenotypes. This comes with some open questions because from 2007 onwards, there are few studies that actually investigated these differences in morphometry. For instance, all participants in the former study were right-handed. And it may be that the shape of this hand knob is somewhat indicative of the handedness. Hence, we can test this by looking at a small data set from Open Neuro of the Amsterdam Open MRI dataset, which collected up to 46 lefties. However, this can also be translated later on at later stages to bigger data sets, such as the HCP. However, in order to do so, we need to come up with a pipeline for analyzing these morphological differences. Hence, one can think about best practices or an automatic pipeline for brain extraction, for the segmentation, and potentially for identifying what I would call a gyri skeleton of the brain. And depending on who is going to join the group and the expertise that we got, we might even think about possibilities for machine learning based classification of these different shapes, because obviously looking at 2000 brains manually is a bit laborsome, and it might be a good thing to automatize this in some way. But another thing that also tackles or is in line with what Daniel talked about his gradients would be to actually identify the high dimensionality and low dimensional space of this gyri skeleton. So it might be that individual differences of different groups like males and females, left-handed, right-handed, become apparent when we embed this complex gyri skeleton into a low-dimensional space. For this project, we could use different tools. For instance, I've played around with it for a bit and created a gyri skeleton simply by using FSL, but some other folks may find it more useful to use FreeSurfer for that. And it would also be great to, in the end, get a Python openly accessible code out there in order to get this pipeline running for other collaborators. And with that, the only question I've got left to ask is who wants to join, who is up for the task. And we're going to start with a brainstorming session around 13:30. Thank you very much for your attention.
Thank you very much, Patrick. You totally made a point. Now, time for project number 4, Marco Marino.
Yep, here I am. Can you hear me?
We can, and we can see you.
Okay, so I share my screen. Just submit this one. Oh, presentation. Nope. Right. Okay. Okay, good. Can you see it? Yeah. Okay, good. Yeah, well, so I'm Marco Marino. I'm a postdoctoral fellow at the KU Leuven in Belgium. And today I'm proposing actually this project, which is something I've not been working on actually. So it is a bit an explorative thing that I want to propose. It is about studying the heart-brain interaction and trying to see if actually it is possible to define a framework in which brain activity, the behavioral aspects, and the peripheral physiology, including ECG recordings, for example, can be linked together and see what is the effect on the large-scale brain networks. So concerning the interaction, there are studies which showed already that the heart and the brain interact, and they affect the behavioral performance for different tasks. Commonly, it is studied the heart rate variability, so in which way our heartbeat changes over time. And it has been shown an increased cognitive performance for different tasks together with the increased heart rate variability. Though I find very interesting, actually, I'm quite passionate in studying the brain during the resting state. So it is this condition in which basically you are not thinking about anything particular. And so you are resting, literally. And so there was a study actually about also Tibetan Buddhist monks meditating. And so they could check actually that there was a link between their heart rate variability change that were associated with the meditation and the default mode network, for example. So I would like to focus on the resting state networks, which are these ones. So for the ones who doesn't know, commonly, I mean, they were first detected with the fMRI, and they basically have topological pattern which recreates the one of the networks that are activated during a certain task. So for example, there is sensory motor, commonly those areas are activated during a motor task. So the idea is to check these large-scale networks and see how they correlate with the ECG recordings, basically. And the idea, actually, it is similar to the one that I've been studying about correlating the fMRI networks with the EEG recordings. So I would like to kind of recreate a similar idea to see in which way the heart rate variability, for example, links to a specific network, and see if a specific network is more affected than others by the heart rate variability. And so does the cardiac activity affect the fMRI networks? Probably yes. because I mean, commonly in fMRI anyway, the cardiac signal is something that is considered in the processing of the data. Usually it is a confound that is removed, but we want to keep it. But we don't know how this affects the network. And especially, we don't know what is telling us about the brain state. So the idea is really to check this triangle, this framework of brain network, behavior, and ECG or respiratory variables. So for the working group, so who I'm looking for, I mean, basically for anyone, I mean, with any background, this is something very welcome, because I think everyone can contribute in different way, either from the methodological point of view, either in the interpretation of the neuropsychology behind a specific network, so for knowing also to which network focus on eventually. I would work on MATLAB, and so people, they should know at least some basics for doing that, and we will use also SPM for some pre-processing. For the neuroimaging skills, don't worry, I mean, it's okay also if you have a basic knowledge. I mean, this is actually required, possibly it would help a lot. If you have an advanced background for it, also it would be great, because in this way we could try with some more advanced approaches. And we are going to try to work on these data sets available, database available from the Max Planck Institute, the group Lemon, which is very cool actually, because it has fMRI resting state network recordings with simultaneous also ECG and PPU and other physiological variables. So if you're interested in disclosing something about the mind-brain-body interaction, this is the group for you. Okay, thank you.
Thank you very much, Marco. Before going on with another presentation, I would like to spend some word on this project, because it has been made possible by suggestion and collaboration in a briefing preparatory for this BrainHack. So thanks everybody for making this project possible.
Yeah, indeed, it was really in the spirit of the BrainHack. So thanks a lot for this chance, actually.
Thank you again to Marco. Moving on to the next group, Arianna Menardi.
All right. Let me just share my screen. All right, I hope you're all able to see it.
We are.
So welcome everybody to BrainHack in Padova. The title of my project is actually an inter-individual difference analysis. Are graph properties enough to explain behavior? And I really hope you'll be able to join me in this adventure. So what are we actually going to cover in this working group? It's a way sort of studying the brain in form of a graph. This has been proven particularly useful, and this is why I've chosen this as a technique. The basic knowledge that it's required and that we're going to cover is that we will start with images, neuroimaging data, for which we have applied an anatomical parcellation. From those parcellations, we are able to derive, of course, identify regions of interest, and we extract time series data from those regions of interest in order to define how different ROIs in the brain interact with one another, and therefore to construct what we call a functional brain network. And from this, we will derive what are the core of the project, which are the to perform some basic graph theoretical analysis. I stress the word on the graph because this is a very unique approach to study how the information flows in the brain, how different subnetworks interact with each other, and they overall defined the peculiarities of the individual brain. So how will be rocking science in these 2-3 days of working project is that we will start with some data that I can provide to you. So we will have already functional matrices, which will kind of give us a head start on focusing just on graph measures without having to run through the preprocessing and stuff. I've also gathered some task fMRI data. So we have cognitive scores for a range of various tasks, from emotion to language to memory, which we can decide together which one to use, if you want to use all of them. and of course the MATLAB code to extract graph theory metrics, and which I will share with you and from which we will start working on. The point where we are all going to focus together is how we can relate all those differences to cognitive scores. So what I'm really hoping is that you can help me out in defining a model to correlate graph theory measures with cognitive scores. So what you will need is, first of all, tons of patience. This is my first time leading a working group rather than being part of one. So sure, something will go wrong, just bear with me. As a second, we are going to use MATLAB. So if you have MATLAB installed in your computer, that would be great. As a way to present and get a sort of an overview of how brain networks represent in the brain, we can use two different approaches and softwares. One is CAFI and the one is BrainNet Viewer. They require different levels of expertise, so we can decide on whether to use both of them, only one of them, or if you have any other suggestion on which one you would like to use, I'm very open to learn also new softwares. So what you can expect as a pro, I think this is going to be a good learning experience for basic graph measures extraction. I think it's also very cool because I think we're going to be able to share a lot of ideas on how to do that. You can get your hands on some codes, which is always good. It's what students are sometimes after. So personally, I find this very useful. You can definitely prove how good you are with MATLAB or how bad you are with MATLAB. That means that there are actually not prerequisites to join us. Whatever it's your background, you can come in. Everyone is welcome. And I'm sure that even if you've never seen MATLAB, we can sort things out and walk you through. And it's also a nice way to understand which are softwares that can help you actually draw some nice figures plot, which you never know for publications, but also to get a sense of the data just by visual inspection. They're very useful. And lots of more pros, but I think that overall it's very good opportunity to teach each other. So I really welcome anyone's feedback. So please. What are the cons? Once you join the dark side, there is no going back. Just to say that there are no real cons. So please join me. I'm really looking forward to see you all. And thank you.
Thank you very much, Arianna. Now moving on, next one is the sixth group. It's a sixth project proposed by Lorenzo Pini.
Yep. Can you hear me and see the screen?
Yes.
Okay. So good morning, everyone. I'm Lorenzo Pini. I work here at the University of Padova, the Padova Neuroscience Center. And the title of my project is the association between the variability in atrophy and the connectivity in Alzheimer's disease. Well, as you know, Alzheimer's disease is one of the most common neurodegenerative disorders affecting millions of people worldwide. We know that there is a cascade of events in the pathophysiology of this disease, starting from the accumulation of amyloid and tau proteins, which leads to atrophy and then to cognitive deficit, where memory is the clinical core. And we also know that these abnormalities start several years before the onset. Here in this table, you can see that the zero, the red column is the onset of clinical symptoms, but we can detect abnormality in several biomarkers, for example, in amyloid, even 20 years before the clinical onset. And then also neurodegeneration can be detected 10 years, and then some years before the onset, we can detect hippocampal volume degeneration and also subtle cognitive deficit. And here in the left, this is the most famous model of these biomarkers in Alzheimer's disease by Clifford Jack, published in 2013, showing this progression. Now, when we, in this progression, in these biomarkers, for example, we know that there is a typical pattern in the atrophy in Alzheimer's disease, which is the trajectory is typical, and we have atrophy starting from the temporal lobe and then following a temporal parietal frontal trajectory, as you can see here. These are patients in a mild stage where the atrophy is predominantly temporal lobe. Where we move in the clinical course, we can see that this atrophy spread also in the frontal parietal and so on. But this is a general scheme. And there are other biomarkers. There are new biomarkers that are developing, which seems promising, which is indeed cognitivity. And we know, for example, that there is, well, it's still under investigation, the time onset in this model of cognitivity abnormalities. But what is interesting and important in this brain project is that there is abnormalities in the hormone network, and abnormalities in the connectivity within this network is linked with amyloid deposition and metabolic disruption and atrophy, which are a marker of neurodegeneration. And this is the general scheme. So we have this temporal parietal frontal trajectory in atrophy and the hormone network abnormalities. But Alzheimer's disease, there is variability in the clinical phenotypes. For example, in this study by Koppel, published in Human Brain Mapping, they show that different phenotypes of Alzheimer's disease have different patterns of atrophy. For example, in red is the early onset Alzheimer's disease, which are patients when the clinical symptoms start before 65 years of age, and which are memory is quite preserved, but they have a deficit in executive and attention functions. While in yellow is the late onset Alzheimer's disease, is the typical Alzheimer's disease. And here, for example, we can see that in the mild stage, late onset, so typical Alzheimer's disease have atrophy in the hippocampus, while, for example, early onset show atrophy in the parietal, temporal, or the blue is the language variant of Alzheimer's disease, where we have a predominant atrophy in the left temporal lobe. And then we have also posterior cortical atrophy, which is the visual variant of Alzheimer's disease, where we have atrophy, we can detect the atrophy very early in the visual cortex. And this different trajectory in the atrophy pattern has been observed also in the connectivity. As I said, there is this pattern in the hormone network. But if we investigate, this is a paper that I published with my previous group, when we investigate the typical Alzheimer's disease, the late onset, we found connectivity abnormalities in the memory networks, the hormone network and the limbic network, which are associated with memory functions. But in the early onset, so which is the disease in which the disease affects especially executive and visuospatial deficit, we found abnormalities in the frontoparietal and also the visual network. So in this BrainHack project, I would like to link these two information. So the variability in the pattern of atrophy in mild AD, prodromal AD, we will use data from mild cognitive impairment. So to link this pattern with the pattern in the functional connectivity. And so the individual profile, both for atrophy and connectivity. What we will do? Well, we start from the ADNI data. ADNI is one of the biggest repository for imaging and genetic data for Alzheimer's disease. I have 165 data from mild cognitive impairment patients with a positive amyloid profile and 123 age-matched controls. So what we need is to extract the gray matter information from the data, and we will use two different approaches. An approach which is called atrophy network mapping. So for every patient, we identify the atrophy profile, and then we construct the networks using a normative data set. And then we will investigate whether this normative data set, so the network constructed in this way, is congruent with the individual network. So we will build the network using the resting state fMRI data of these patients. If you join this project, depending also on the skills, you could assess the segmentation of the image that they have already processed, so quality check. And we also need to extract the seed atrophy to construct the networks. Then, of course, we need to do some data extraction to investigate the cognitive and the molecular profile of these patients, and molecular profile from CSF and PET data, and so from the Excel data that I have. And then, if we have time, if we can do it, we can extract some preliminary functional connectivity data. We are going to use MATLAB, Bash, and FreeSurfer. That's all.
Thank you so much, Lorenzo. Going on to the 7th project, Cristina Carpazza, Enrico Toffalini, and Massimo Grassi.
Yes.
Is
Hi.
anybody- Hi.
Hi to everyone. Okay. So can you see my desktop?
We can.
Okay, perfect. So in the summer of 2019, I won a travel award to participate to the OHBM hackathon. When I was there, I chose to work in the COBIDAS working group. COBIDAS are guidelines that collect best practice recommendations from experts of the entire brain imaging community. These recommendations covers the whole project from the data acquisition to statistical analysis. And I thought, oh, that's very important and might help to mitigate the problem of absence of replication. So far, in medicine, it's quite common to follow standardized guidelines. For instance, in recent years, stroke guidelines have been created to report observational studies, care guidelines to report case reports, STRIGA guidelines to report genetic association, CONSORT guidelines to report clinical trials, PRISMA guidelines to report systematic reviews and meta-analysis. These are, of course, only examples. Surprisingly, nothing so far of similar is available in the scientific literature to report cognitive and neuropsychological studies. So the aim of this BrainHack for me, my colleagues and I would like to propose to work on a first draft of the pre-CAMS guidelines to report studies in cognitive science and neuropsychology. The aim of the pre-CAMS guidelines will be the following. First, they will aim to help scientists to plan a new study. Second, they will aim to guide how to write a scientific paper and ensure that all the necessary information to allow replication of the results are available within the paper. Third, they will help to evaluate the presence of bias in previous studies. Indeed, the risk of bias assessment is one of the items requested by the PRISMA guidelines, but so far, no instrument to guide risk of bias assessment for cognitive neuroscience is available. So to conclude, to summarize, the pre-CAMS guidelines will aim to be a very valuable instrument to help to mitigate the problem of replicability in cognitive psychology and neuropsychology. So we are not going to analyze real data, but we are going to think on what is important and needs to be replicated and to create these guidelines. Thank you.
Thank you very much, Cristina. Now, if I can hold on a second, and it's time for the very last working group proposal by Nicolas Trevisan. Nicolas, are you here?
Yes, now you can hear me, right? Okay. Can you hear me?
We can hear you.
Perfect.
I cannot personally see your face in this moment.
Yes, we can see also the face, yes.
Perfect. Okay,
Okay.
so I'm going to share my screen. Should be okay, right?
It is.
Okay. So my project, so I'm Nicola Trevisan. I'm a PhD student in neuroscience here at the Padova Neuroscience Center. And my project is called Brain Morphometry in Chess Player. What do you need to be a Grandmaster? But it's because I choose to download 58 anatomical scans dataset that actually are composed by 2 different groups. The most important one are the 29 chess masters. Chess masters are very skilled chess player. In this case, are actually all Chinese people that are super good at the international level. So actually quite famous people looked around. And also we are going to compare them to 29 control. So normal people with little or no expertise on the chess game. we want to investigate, I want to investigate a difference in the structural brain between the two groups. And hopefully I think we can find something because, you know, chess players tend to be like very skilled. I think they have something more than normal people that can explain this difference in skills. To do that, we're going to use SPM, which is basically the most famous software for statistical analysis of magnetic resonance imaging. And we are going to analyze the difference in gray matter volume and white matter volume, that is the voxel-based morphometry. But also we want to investigate the surface-based morphometry, which is basically analyzing other indices other than the gray matter and the white matter. That is the structure of the surface of the brain, of the cortex, such as the gerification index, the cortical complexity, cortical thickness, and the cycle depth. To do this surface-based morphometry, we're also going to learn how to use CAT, the Computational Anatomy Toolbox, which is a toolbox inside SPM. It's also super useful to extract all these surface values and analyze them. I also prepare a timetable to show you how these 3 days, if you decide to join my group, can get out. So the first day, today, I want you to install a viewer to navigate, explore anatomical scan. So for all the people who actually maybe never been able to work or just handle raw data, raw anatomical scan, that can be super useful. We're also going to install SPM and CAT, the two toolboxes, the two software we're going to use. And of course, we're going to start with handling this anatomical scan by align all these raw data so that SPM can actually understand them a little bit better. And the second day, we're going to start with the preprocessing. The first step of the preprocessing is the segmentation, which means that we are going to segment, so divide the brain between the gray matter, white matter, and all the other surface data that I showed you before, like the gerification index or the cortical thickness. Then to all these segmented data that we got from the first process of the preprocessing, we're going to apply a filter. So to smooth all those data, those extracted data, then we're going to take all these data that we gather with the preprocessing, and we're going to do a statistical analysis. Being this a two-group scenario with the chess master and the control group, we just need a simple t-test. Finally, on the last day, this is the day where we're going to collaborate the more, I think. We're going to display the results. Hopefully we can find something, but I think we can. And create the images, and then we're going to discuss the results. That is, we're going to find something, find some difference in the brain, and the most important bit will be to make some sense out of it, right? So if we find some differences in the brain of this chess master, or the other way around, maybe if there's something less than the control group, what does it mean? Why does it reflect? Does it reflect on the ability, on the skill to play the game? Does it reflect on some other data? So we can try to make some sense out of it. And lastly, we can prepare a PowerPoint to share our super interesting results with other people here at the BrainHack. And I think this is a general overview of what we are going to do. Just a final warning, you need MATLAB because SPM and CAT are all MATLAB based. You don't need to be proficient with MATLAB. It's completely fine if you don't know how to use it. That is because we're going through every step together. So it's not a problem if you don't know how to use it, but you still need to use MATLAB to install all the other things. And that's it. I think that I told everything I did. I think we can go to lunch too, right?
Thank you, Nicola. We sure deserved a lunch break. But before that, great news, because Emma, the speaker of the GitHub tutorial, has proposed, and this is very good news. We're really We're really happy about it. To help novice GitHub users with the session. So if you're not proficient or you're novel in GitHub using, she can provide a session to help you.
Yeah, I would like to say a couple of words. These are really great themes for the working groups. There is a lot of complementarity. We range from functional MRI to anatomical neuroimaging, neuropsychology, cognitive and clinical neuroscience, so a lot of multidisciplinarity, etc. I would personally be very confused if I have to pick up one of these working groups. When organizing this event with Davide and Georgia, Davide forced us to give a 3 days brain hack. I thought it would have been too much, but now I would like to have this brain hack with projects in series rather than in parallel to allow us to take part of many of these.
Like a week brain hack. The brain hack week.
No, I would think about months. These projects are so great. Anyway.
I'm really impressed by the level of all the presentation. I'm seriously impressed. So it's promising and good job and good luck. I remind you that we will see at 4 o'clock for the Ignite talks, and the Zoom meeting link is not this one, but you can find the link on the PNC website. At 1:30 o'clock you will join your working group, so you have 1 hour to eat and to decide which project you want to join. What else?
Yeah, the Zoom links to each working group are already available, I guess, right?
Yeah, yeah.
Everyone is.
Yeah.
So lunch break for everybody. That's gonna be probably your hardest hour ever because you have to decide only one of these groups. So happy lunch, everybody. Happy brain hacking. Thanks for joining us.
Thank you.
Thank you.
Bye.
Thank you. See
See you
you
later.
Thank you.
I shall now stop the recording. 1, 2, 3.